So there are four objectives of Nipah virus of our presentation. Firstly, to understand the history of Nipah virus. Secondly, to acknowledge the properties of Nipah virus. Thirdly, to identify disease it causes and the prevention measurement. And last but not least, to learn the laboratory detection for Nipah virus. Next slide. So, firstly, we are going to talk about introduction. Slide. So, where does the name of Nipah virus come from? So basically, the Nipah virus name came from the first village that the virus struck near Kuala Lumpur, Malaysia, that was discovered in 1999. The Nipah virus is similar to Hendra virus that was discovered in Australia 1994. It is paramyxovirus in genus Nipah virus. Nipah virus come from different variants were involved outbreak in Malaysia, Bangladesh, and India, which are two major strains isolated from peak in Malaysia. There are two causes of Nipah virus. Firstly, severe rapid progressive encephalitis in human, which are highly mortality rate for human. And lastly, severe respiratory illness in pigs, which are some demonstrate nervous system sign. The transmission of Nipah virus to human is when close contact with infected pig. And Nipah virus survive in environment for long period, which is in a favorable condition, for example, for days, in fruit bag urine and contaminated fruit juice. Next. So, next we are going to talk about properties of Nipah virus. The Nipah virus is a type of RNA virus in the genus Nipah virus. The virus normally circulate among some fruit bats, and lastly, it can both spread between people and from other animals to people. Next slide. In addition of properties of Nipah virus can infect a large variety of mammalian species. The structure of Nipah virus is a long, parallel tetrametric called with a small alpha helical cap. And last but not least, Nipah virus is not an airborne infection. So I will pass the next presentation to Intan.
Okay, thank you, Fitri Akil. So we move to the next topic, which is effects of Nipah virus. Okay, Nipah virus can cause fever, headache, cough, sore throat, difficulty of breathing, and also vomiting. Okay, next, there are 5 ways to prevent of Nipah virus. The first one is practice hand washing regularly with soap and water. The second one is avoid contact with sick bats or pigs. The third one is avoid areas where bats are known to roost. The fourth one is avoid eating or drinking products that could be contaminated by bats. And the last one is avoid contact with the blood or body fluids of person known to be infected with Nipah virus. Okay, next, there are 2 biochemical tests that we can use to detect Nipah virus. The first one is catalase test. Catalase is an enzyme that breaks down the toxic chemical hydrogen peroxide into water and oxygen. And the second test is oxidase test. The oxidase test determines whether or not an organism has the cytochrome oxidase enzyme. Okay, so next we move on how to diagnose the Nipah virus. Nipah virus infection can be diagnosed with clinical history during the acute and convalescent phase of the disease. The main tests used are real-time polymerase chain reaction, which is RT-PCR, from bodily fluids and antibody detection via enzyme-linked immunosorbent assay, which is ELISA. Okay, why PCR is recommended as the most sensitive method for diagnosis of active Nipah virus infection? Nipah virus specific IgM, ELISA, is an alternative serological approach where PCR is not available. Other tests used include PCR assay and virus isolation by cell culture. In addition, the quality, quantity, type, timing of clinical sample collection, and the time needed to transfer samples to the laboratory can affect the accuracy of laboratory results. Nipah virus infection can be diagnosed during illness or after recovery. Different tests are available to diagnose Nipah virus infection. During early stages of the illness, laboratory testing can be conducted using real-time polymerase chain reaction, which is RT-PCR, from throat or nasal swabs, cerebrospinal fluid, urine, and blood. Later in the course of illness and after recovery, testing for antibodies is conducted using an ELISA. So I think that's all from our group. Thank you so much for listening.
Thank you, everyone.